Hello all! The subject of this video is to explore the Human Genome Project and explain the techniques used, the benefits and limitations of the project, and an explanation of genetic linkage maps. In the bottom of the screen, you'll often see some text, and that's just a reference to the intellectual owners of the images I'll be using. And first up is this relatively basic and generic timeline of the project itself. Initiated in 1990 and fully completed after 13 long years of government-funded research across 18 countries, the Human Genome Project successfully mapped the entire human genome, resulting in an entire sequence of 3 billion base pairs. A parallel project was conducted by Celera Genomics in 1998 and made heavy use of its advanced sequencing algorithms and technology to match the progress of the government-funded project. In the February of 2001, the International Human Genome Sequencing Consortium published its first draft, which suggested that significantly fewer genes than expected, with about 250,000 genes, were needed to complete the human sequence. The primary method employed by the Human Genome Project from 1990 to 1980 was an annotative method that involved the bacterial artificial chromosome method, or BAC method. To achieve the end goal of a chromosome sequence, the chromosomes were divided up into relatively large portions of 150,000 base pairs. Following this, they were cloned in a bacterial cell by using recombinant DNA principle. Next, the sliced up genes were fingerprinted through gel electrophoresis and annotated by the analytical biologists, according to the weight of each base. Alternatively, the sliced up genes could be further divided into base pairs of about 500 and sequenced through a program called PHRAP, which searches for common overlapping introns. This process can be likened to chopping up a book, transcribing many copies of that book and reassembling it by looking for sentences that overlap to make inherent sentences, with repeating introns being the overlapping sentences. Unfortunately, this process is quite time and labour intensive, which caused the early years of the project to pass at a snail's pace. However, with the introduction of the whole genome shotgun sequencing method in 1998 by Celera Genomics, many of the steps previously mentioned could be bypassed entirely, as shown in this diagram. For starters, the genome is shredded into strands about 2,000 base pairs long, and once again to form some that are just 10,000 base pairs long. Similar to the BAC method, these pieces were cloned in bacteria and then sequenced and assembled through a gene matcher processing unit and many terabytes of storage. In spite of its late introduction, the shotgun method accounted for 90% of sequences yielded by the project. Hopefully this graph will aid in understanding the overwhelming benefit of the shotgun method, as progress increased exponentially with the introduction of Celera in 1998. Advances in computing and technology were what ultimately made this leap in methodology possible, as well as private enterprise interest in biotechnology. The Human Genome Project accounted for benefits not only within its own field of biology, but also influenced the way in which genetic research would from then on be conducted. The immediate and most obvious benefits of the Human Genome Project include new avenues for the development of medicine and technology, where tests for genetically predisposed conditions such as breast cancer, cystic fibrosis, and liver disease. Areas such as cancer and Alzheimer's research are also expected to benefit from the project, as researchers will be able to trace among both introns and extrons for common markers and/or defaults in the entirety of the human genome. Likewise, the human genome sequence will aid in understanding the function of proteins and determining evolutionary paths. Unfortunately, the initial hopes of accelerating genetic disease research was not entirely achieved by the Human Genome Project, as more is required than simply the sequence of the human genome. Consider how an architect may look at a master plan for a house and determine where the main support beams and foundations would lie. The human genome acts more as a toolbox, where all the tools necessary to build that house are there, but the tools are selected by the internal state of the organism and by the state of the environment around them. Similarly, the understanding of genetic disorders had previously been that it was one gene to one disorder. Yet now it is known that a variety of genes or environmental factors may affect the prevalence of a genetic disease. For example, the identified breast cancer gene accounts for only 10% of cases, unlike cystic fibrosis, which is characterized by one gene and is therefore less complex. Having the genome sequence does not necessarily guarantee that effective treatments will be developed, though it is one step closer to doing so. When discussing genetic linkage maps, I should first mention that it is not a physical map derived from radiation-reduced hybridization, or a gene map. Instead, it is a map derived from a population or species, regarding genes' positions relative to each other in terms of recombinant frequency. That is, it is based on the frequencies of recombination between genes, whilst homologous chromosomes are crossing over. Hopefully this diagram from the University of Utah will make things a little bit clearer, as linked genes such as gene 1 and gene 2 in this diagram are more likely to remain together during the crossing over period. For this reason, linkage maps are critical for locating genes associated with genetic diseases, as linked genes are more likely to be expressed together. Therefore, if one gene's position is known, researchers are able to use the relatively small distance between the linked genes to determine the faulty gene's location on the chromosome. In this picture, we can see that the closer the genes are on the same chromosome, the less likely they are to be separated, and so are generally expressed together. However, as I have previously mentioned, linkage maps rely on knowing the location of one gene in a linked pair or group, in order to determine the position of the faulty gene in question. Linkage maps may only provide knowledge based on previous knowledge derived from base-by-base research, much like what was done with cystic fibrosis gene that required years of chromosomal scouting to locate. In this way, linkage maps are very much a causal method, relying on what is already known. Likewise, the complexity of identifying genes is beyond what a linkage map is able to provide, largely due to the overlapping and repeating segments, and the high level of variability associated with crossing over. It should also be noted that linkage maps provide the location of genes in proximal terms, as opposed to definitive mapping, and must be narrowed down through physical mapping and genetic identification. Therefore, when starting from near scratch to map the human genome, linkage maps are an inappropriate and ineffective method for the Human Genome Project. Just to recap what has been said throughout this video, the Human Genome Project began in 1990 and finished in 2000.